Thanks for the invitation and thanks for everyone for coming. And I'm really happy to share the airborne work we do here. And before I get started, I want to say all the work I presented here is a kind of teamwork. So it cannot be simply done by one single person or two people. So it's a huge work and built on a long history of airborne work we do here. So let's get started. The first thing, let's solve some technical terms first. So imaging spectroscopy. Spectroscopy, I asked my wife last night, I said, do you know what is spectroscopy? And she said, yes, but I don't know. And I heard you talk about it all the time, but probably you need to explain that a little bit. I say, okay. So why it doesn't want to go? Yeah, here we go. So spectroscopy is actually a very old technique. So a few hundred years ago, Newton split light to different colors, like the rainbow color, using his little prism. And that he also opened the door for a new technique we can use that's called spectroscopy. Basically, let's say if you throw a light or white light into like kind of a material, and then you use the reflected or absorbed light to study the target material. And that can be called spectroscopy. That's a few hundred years. And there has been a kind of mature technique people can use both in science and also industry. So we use that in remote sensing. So I copied and pasted a definition from a paper here. And you may say terms like spectroscopy, imaging spectroscopy, and hyperspectral imaging, and some people can call that ultraspectral imaging. Sometimes these terms can be used interchangeably. And here it says the simultaneous acquisition of images in many narrow spectrally continuous bands measured from a remotely operated platform. So basically we got an image. So here's the image. And for each pixel of this image, we are not only getting the three typical bands we can get to use cell phone or camera, red, green, blue. Here, we get hundreds or thousands of bands, continuous bands, like very narrow wavelengths or very narrow color. Some we can see the visible color from 400 to 700, we can see with our eyes, and also some wavelengths we can't see, but we can design an instrument to measure that. Then we can use this to study the landscape in this case. So imaging spectroscopy, we get image, we get spectral, put two sets together. That's it. Second thing, remote sensing. So when we say remote sensing, if we use a broad definition of remote sensing, anything we use to study or to learn the characteristics of a target without in contact with the target, we can call it remote sensing. Like we use our eyes to see things. That's remote sensing, right? It's very well-designed sensors, actually. So here, we have different satellites. We have aircraft, UAV, drones. We have all kinds of ground sensors. This is all part of remote sensing. But for the Earth observation today, I'll focus on the sensors here, those in the air, and they rely on the electromagnetic energy. So we measure reflected or emitted energy from satellites, aircraft, UAV, drones. And in particular, I want to focus on aircraft today in my talk. Then why aircraft? So there's a book published by the National Academies of Science, Engineering, and Medicine in 2021, so last year. Actually, they had a meeting during the COVID time, so it's kind of a virtual meeting. It was fun to read this book. So this book was published in 2021, and they have a huge group of scientists, and they put these people together and to discuss one thing. to airborne platform to advance NASA Earth system science priorities. And they were discussing, is there a need to include a new large airplane, or even to maintain the large airplane in the NASA current aircraft fleet for the future scientific research? Like after 2 days discussion, and they reached a consensus report. And here's one of the conclusions I copied from the report. It said, while there are some promising developments of drones, balloons, and these technologies may not advance quickly enough to contribute significantly to Earth system science research with the next decade, well, we have seen a huge development of using drones, UAS, or UAVs to study the Earth. Well, we also have a few drones here in this building, but it sounds like the aircraft can play a unique role. We still need this thing. So today, I want to share some work we did or we are doing using the aircraft we have here. So we're not building from nothing. Actually, this aircraft study or the hyperspectral imaging, we put that sensors on the aircraft starts like 20 years ago. So this paper was published in early 2007. And I don't know if that's the earliest paper published using this early image spectrometer, the ESA Eagle. So, but that's the earliest paper I found. So 15 years ago, we have this aircraft. We put a hyperspectral imaging spectrometer on this and fly all kinds of ecosystems, including here, the coral reef. That's CALMIT. But we can't stay there and say, we have this system and we have been great. We need to do some new things, right? We need to improve from what we have been done from the past. Then we build the Nebraska Earth Observatory. So we have a new name, NEO. And that's an old aircraft, but we put some new instrument into it. So it's like you add some new one to the old bottle, but it's a new one, right? So that's a new airplane. And we have a few new instruments. When I say we, I mean the whole team of the whole CALMIT team with the support from SNR and the whole UNL community. We have a IBIS spectrometer, we have a Kestrel, so we have a thermal sensor, but it's not here. We can sense the visible light. So from 400, 700, the light we can see with the eyes. We can see the near infrared, part of the near infrared, this region. And we can see the thermal refract, this thermal energy. One special thing about this set of new instruments is about the IBIS. It can be used to detect a very tiny signal called solar-induced fluorescence here, SIF. So this energy is very, very tiny, and it's about 3% of the illumination. And we'll come back to this in the next few slides, but I want to keep everything in one single slide here. Okay, we have this new toy. How can we use it, and what we did with this new tool? So here is the Nebraska, and I copied this figure from the large map of precipitation change during the last 30 years. So there was a news published by the New York Times last year saying part of the country is getting wetter during last century and the last 30 years. And then part of the country is getting drier. And we're in Nebraska, we are in the middle. See here, there's kind of a line in the middle. So when it gets greener, that means this part of Nebraska is getting wetter. And when it's in yellow, this part of Nebraska is getting drier. So because we are in the middle, we don't need to go outside of the state to study how the climate change affects the ecosystem, different ecosystems. We can stay in Nebraska and study this whole thing. So it's kind of lucky to do this. And today I'll discuss the 3 kind of ecosystems we do. So agriculture, forest, prairie. So agriculture is really hard to stay away from agriculture when you are in Nebraska, right? We are the corn husker state. We have to talk about the corn and soybeans. And forests, there are not many forests in Nebraska, but there are some, and they play important roles here. And we have large amount of prairies. So the yellow points are prairie size we have. So let's start from the agriculture. So photosynthesis of crop fields. I've been showing this figure multiple times, and it's a product we obtained in 2018. We had, I forgot how many flights, maybe around 10 field campaigns, airborne campaigns in 2018. And we got a map like this in MID, NRAC MID, for different flight size, different field size. So each of those yellow dots represent a flux tower. So by combining the airborne data, the images, and the flux towers, we can study the photosynthesis at different scales, time and spatial scales. So typically when we say photosynthesis or say remote sensing, the first thing comes to mind is NDVI, right? That's the index developed 30 years ago, and it's still very useful. And chlorophyll indices. So that's actually developed by Gittelson here. It's worked like 20 years work on the chlorophyll indices. It has been widely used now. And the idea is very simple. Let's say there are two different targets, a spectral of different targets. One is the vegetation and one is the bare soil. You see here, this part is the red band, this is the near infrared. Because of vegetation, it doesn't need the light in this energy, so they reflected a lot of energy in the near infrared, and we utilize this difference in reflectance between vegetation and the soil in the infrared and the red band, and calculate the normalized difference. That's the NDVI to separate these two, and the NDVI can be used as the indicator of the greenness of the target. Oh, it's a similar idea. Instead of using near infrared and the red band, we use the red edge, which is the sharp increase between the red and the near infrared of the vegetation reflectance spectral. So we use that and relate to this index to chlorophyll content at different scales from a single leaf to the canopy, right? Then we can apply these simple equations to the airborne data. Then we get images like this. NDVI image, chlorophyll. This one is one of the chlorophyll indices. This uses a red edge, so it's called the chlorophyll index red edge. So we calculate the NDVI image, we calculate the chlorophyll index image. Cool. These images are pretty. High values means higher NDVI, high values means high chlorophyll. But we have been doing it for 20 years. Nothing fancy, nothing new. I'm not gonna spoil you guys with 20-year-old data or products. Let's move a little bit of one more step further to see what we can do with the new technology. Okay, let's step back a little bit, get back to the theory of photosynthesis. I don't think I have enough time to go over all those details, and to be honest, I don't know I fully understand all those details here. But to keep the thing simple, when the plant gets the light, there are three ways the light can go. One way is start the electron transport, so let's go to the photosynthesis. Or it's go to chlorophyll fluorescence. It's a tiny signal during the fluorescence, during the photosynthesis. And then the third pathway is going to become heat, release as heat. And this figure shows the whole electron transport in the photosynthesis. And we can see that the chlorophyll fluorescence here is a physical process directly linked to the electron flux. Then we can use this fluorescence to study photosynthesis yield, the plant water stress, nitrogen status, etc. And it's a powerful tool, but it's kind of old. Then that's called for fluorescence. How can we measure it using remote sensing? So let's say here is one, the solar radiation. The black line indicates the solar radiation. And the red line, I call it the plant reflected radiation. We put a sensor above the plant. Let's say measure the radiation. I put this reflected in quotation because actually it has two components. One is the reflected energy, and then the second part is this guy. It's fluorescence. So it's actually emitted energy from the plant. It's not reflected. Then how can we separate these two? Because as I said, the fluorescence is a very tiny signal. How can you separate 3% of the total illumination from the rest? It's hard. Well, see these lines, these dips, particularly this one here. This line is called the Frohoff lines. So this caused by the oxygen here. The oxygen in the atmosphere, they absorb almost all the energy in this band, 760. You see here, the solar energy is very low. Then we can extract the fluorescence using these bands. The idea is, because the solar-induced fluorescence, the fluorescence is very small. Let's think about it this way. If I turn on my cell phone here in this room, because the lights are on, you can barely see it. But if I turn all the lights off and then turn on my cell phone, it's easier to see it, right? The same idea applies to this. So in this band, when all the energy is absorbed by the atmosphere, so a tiny little energy added by the plants, you can extract it here using this absorption band. That's one method to extract the fluorescence. So instead of putting the fluorescence line here, I use the reflectance line. So that's the radiation, the solar radiation. That's the reflected radiation of the plants. And then that's the reflectance value. Let's focus on this region here, around the 760 nanometer. You see the blue line, that's the reflectance line. You see a spike here. That is actually caused by the fluorescence. That's emitted energy. That's the actual energy. And then we can detect that in this area, in this region. But it requires an instrument with a very high spectral resolution to detect the safe signal because it's very, very small. That's why we need this guy, the IBIS. Okay, that's the basic idea of how to extract fluorescence from the remote sensing signal. But the actual processing is kind of very complex. I'm not going into that details, but you just need to know we have a way to extract the fluorescence. That's good enough. Good enough for now. Okay, so here is what we do. We designed this experiment. We put before we can use the airborne remote sensing, the airborne fluorescence to study the plants from the air. One thing we need to know is what we measure from the air is actually comparable to the ground data. That is important. We need to know that. Otherwise, how you can say the thing you measure from the air is fluorescence, is photosynthesis. We need to validate the data, build this connection. So we designed this experiment. We have this detector. It's a fine spectral resolution detector, can be used to extract the fluorescence on the ground. We mount that on this tower, and that's the plant below it. That's in 2018. And we have a flux tower which measures the eddy covariance. That's the GPT, the actual productivity of the plants. That's kind of ground truth, the blue line here in this figure. And then we fly the airplane a few times through the season, and this round circle dots, red, blue, and they are the airborne data. So let's put everything together. We have a blue line, that's the GPP measured using the flux data eddy covariance. We have the red lines. The red line is the fluorescence, solar-induced fluorescence measured by this guy. And we have the NDVI, the typical remote sensing products. Like you need, when you study plants to use remote sensing, you have to calculate NDVI. You just have to. We put these three things together, and you see the first thing is, well, the airborne data is actually comparable to the ground measurements. You see the red line, the red dot, black and black, black line, the black dot. They agree typically very well. That's good. We want to know that because that gives us confidence on the airborne data. So that's something we can validate on the ground. And the second thing is the fluorescence follows the GPP better than NDVI, particularly in the late season. When the GPP goes down, NDVI is still very high, but you see the fluorescence follows this pattern. And if we put the airborne image calculates the fluorescence, NDVI, chlorophyll index for 3 different days in July, August, September, and for the different crop types in this area. And we get a map of this one. You see the fluorescence, it also reveals the very, very fine scale variations of plant photosynthesis in space, these areas, and the time from July, August to September. And if we put everything together, we combine fluorescence, NDVI, chlorophyll index, and many, many other indices we can calculate. It gives us a new way to evaluate or to estimate the plant photosynthesis from the air. So a short summary for this part for the agriculture applications. So in this paper, this authors propose that solar-induced fluorescence is the key to bridge photosynthesis of plant studies from the molecular scale to ecosystem functions. That's the whole scheme they propose in the paper. And to me, I think the airborne data or the airborne image derived fluorescence is actually the glue to stick everything together. So that's the first thing. Second one, forest. Right? We're going to talk 3 different ecosystems: agriculture, forest, and prairie. So the second one is forest. So we use 2 forest experiments, 2 forest sites to do our experiments. The 2 Smithsonian Forest Global Earth Observatory, so the forest geoplots in Nebraska. One is Indian Cave, this one is Indian Cave, and this one is Niobrara. And these plots are installed and maintained by Professor Sabrina Russo from biological science. So these are the hyperspectral images we collected over these 2 plots, and we also have a thermal image for the Niobrara early this year. Okay, let's go back to this figure. This is simple photosynthesis figure. That's probably the simplest photosynthesis figure I have ever seen. We discussed this part, the chlorophyll fluorescence. But what about the plants get too much light? What plants do? You need to release that energy, right? Through non-photochemical quenching. So they release that energy used by heat. People believe that when plants want to start to initiate the non-photochemical quenching, these pigments, violet-xanthine, zeaxanthin, these pigments, the xanthophyll cycle, will be used related to this non-NPQ, non-photochemical quenching. Because this zeaxanthin, it has a high rate of heat loss. For example, let's say you move a plant from low light to high light, you will see these pigments change from violet-xanthine to zeaxanthin. Okay, fine. Then is there anything to do with remote sensing? Fortunately, yes. go back. Okay. So we have an index called the photochemical reflectance index, PRI. It was developed by John. It's sitting somewhere in this room. So if you want to know how he came up with this idea to have an index to monitor this pigments change, you can go to talk to him. That's like 20, 30 years ago. But now we do have an index. We have PRI to actually evaluate or estimate this whole process at different scales from leaf to canopy, because there's like 20 years of research on this topic. And it's clear that this PRI is affected or influenced by the incoming light, right? Because when you have actual light, actual energy, then this whole processing will be initiated by the plants to release this actual energy. Then how about to put the light and the PRI in the landscape scale? So that's the Indian cave plot. So it's somewhere in this area. And we calculate one index called the power albedo, which is an indicator of the incoming light at the pixel level. And then we calculate the PRI for this whole area, same area, and we put them and link them together. And here we use our imaging segmentation or image classification to distinguish different tree crowns and different tree species. It's here because if you have been to Indian cave, you can try to think about that. It's really hard to identify every single tree and to allocate a species to that tree crown, because some tree canopies, they just get together. It's really hard to separate them or impossible to separate them. So in this work, we find about like 1,000 trees that we can identify them, the species of them, and to draw like a polygon to extract that tree crown. And then we calculate the average PRI and the power albedo, which is incoming light. And we put that in a 3D map. So you see this here, there's a valley in this landscape, and we have some ridge here and another 2 valleys here. And we have higher sunlight or higher illumination on top of the ridge, and the lower illumination down bottom of the valley, right? That's right. And we have lower PRI values on top of the ridge, and here higher values down in the valley. And we link these two by tree crowns and by species. And I'm using 3 species as an example to show that actually these 3 species, they have different PRI to light responses. You see the red, the green, this purplish, this yellow lines, they are actually 3 different species. And if we zoom into each of these, that's the purple one here, that's like a blue to grayish line here, and that's a yellow line. These 3 species are different. And we think we are seeing the variations among species. The PRI to power albedo light responses varies by species, and is also affected by the landscape. You can see the topography here, and you can imagine the light response of trees, even the same species, will be different from the top of the hill to bottom of the valley. So this is the ongoing work, and the paper is still on the review. And I would say this whole work is kind of very complicated, and I skipped a lot of details discussed here behind the scene, behind this single figure. So if you are interested in this figure, in these results, we can discuss that more afterwards. So besides this natural landscape, we also studied the trees on campus. Why? Because they are there and we are on campus, so it's easier to study these trees. Kind of find a good excuse to not go into the field. So we have these campus trees, and we have image, airborne image. From this image, it said, well, there are trees, they're green, they're more or less identical, at least to my eyes. And we got a data set, like the GIS people on campus, they maintain a data set for different trees. They have a species identified for each single tree. So that we can use that data set to do an image classification using the airborne data. We identified that 12, I think it's 11 different species, like the dominant species on campus. And this classification accuracy is about like 85 to 90%. It's pretty high actually, but this application is an easier version for tree identification or classification compared to the natural landscape. But it's still not bad at all. But again, image classification using airborne data, nothing new, has been there for 20 years again. Then what about we add an actual layer? We add a layer about state, fluorescence, to this classification to study or to monitor the photosynthesis of different trees. I want to point at this area, you see the red values, that means a higher safe, higher photosynthesis. And we compare this cluster of trees to the trees here. That's these guys. Clearly, these trees were happier than these trees standing in the center of the parking lot in the hot midsummer day. So we're still trying to study this data set, but these are the preliminary results we got. So the second summary, the summary for the second section, we think we are seeing or developing a new way to remotely access the dynamic functional difference in photoprotection and photosynthetic activity in complex stands, either in the natural landscape and also for this urban forests. And these new tools can be used to assess the functional diversity for landscape and to provide the visual indicators of photosynthetic downregulation in response to environmental stressors like drought. Right, in this area, we'll expect less water availability on top of the hill. So the third one, priory. So I'm using some results from early this summer. I'm still actually digesting this data, this result. I got this results last night. So we use the data from Dave Dean's bobcat priory restoration experiment. I didn't see Dave here, but he actually had a very complex experiment. I had a hard time to remember all these things, which is which. So I try to summarize that in an easy way to understand. So basically, it has two treatments. One is burn, the other is hay. So all these plots in the white rectangle, they are burn plots. These burned plots, the first time they burned these plots were last year, May 10th, 2021. And these blue lines, so these plots without these rectangles, they are hate plots. These plots were hit annually starting 2019. So in August, once a year. And you see they have different colors. This yellowish, pinkish, green, and I summarize it here. So it's also a kind of different treatments. The pinkish, it has the seed is selected from 150 species seed mix, and it's herbicide drill. And the yellow is 40 species seed mix. Also herbicide discard and broadcast. And the green ones, these green ones, they are control plots. Nothing has been done to these plots. So remember that this experiment started in 2019. So in the first year, August 6, 2019, we flied over his plot and we got an image like this one. And this is a false color image with the near infrared band go to the red channel, and then the red band go to the green channel, and the green band go to the blue channel. So all the vegetation becomes red in these images. And also we flight it this year, July 20th. When I put these two images together, can you see a big difference? Well, to me, I stared these images for I don't know how many minutes, but the most obvious difference I noticed that there are some trees in this corner, they grow bigger. That's what I saw from this image. But if we calculate some indices from this data, first one, NDVI. Okay, red color means higher NDVI, yellow means lower NDVI, or the blue means lower NDVI. So that's 2019, and this is 2022, early this summer. And we do see a difference, right? You see here, high NDVI, and this part is getting lower. And this plot, the NDVI value is actually very low from this year's data, but it was not the case in 2019. And then we calculate another index called spectral diversity using a moving window. So when you see this figure, so first this one evaluates like when the value goes high, which means the orange color, it means there's a higher spectral diversity. And we can link that to the ground species diversity, which typically we can treat that a higher diversity on the ground. That's a hypothesis. But when you see this figure, you say, okay, these trails or the dirt roads, they have the highest spectral diversity. That doesn't make sense because this figure is actually generated with a moving window. So 10 meter by 10 meter moving window is this big from the left top corner, and it changes. And when it goes to the third row, this pass, this value, it makes the pixels from this part plot, and the road. And this trail is different from the plot. So obviously it will have a higher value, but let's ignore them for now and focus on this blue or like less reddish plot areas. You see here, this plot, it has very low spectral diversity compared to this one and this one, right? And we see a huge change in these spectral diversity patterns from the first year to this year. And then let's put all the data from this year together. So that's the experimental design. That's the three types of minor scale managements. And the first color image, this one is NDVI, spectral diversity, and the thermal, that's the temperature. Let's use these three plots here. Oh, by the way, these numbers, these bolded numbers in the plot, that means the species richness sampled in 2021. That's the last summer. So simply say here, this 2, that means there's a 2 species plot. And here, this one is 41, so this plot has 41 species. And we use 3 plots. One is the low diversity, the intermediate level of diversity, and the high diversity plot. We put these 3 together. See, NDVI, this one low diversity, very low NDVI, high diversity, higher NDVI values. If we use NDVI as a productivity, that kind of makes sense. So lower diversity plot has lower productivity. And if we use the spectral diversity, see this one, the low diversity, the low species diversity plot has lower spectral diversity compared to the high species diversity plot, right? These 2. And when we add the thermal data, you see this plot, it has higher temperature. And this high diversity plot, the temperature is lower. I put everything together, we got this relationship or kind of a trend. When we have a high species diversity in the plot on the ground, we get high spectral diversity, and we get a large NDVI or higher productivity, and we have a low temperature. But the management schemes, either you burn it or you hate it, or all the other management, they affected the diversity and the productivity. And it seems that all these things here can be detected using airborne remote sensing. So again, this is the data we just collected this summer. There's a lot of more work needs to be done to interpret the data. And we have a lot of work to do in the future. So some take-home messages for the talk today. The airborne remote sensing is a unique and a powerful tool to monitor the changing Earth. So we use 3 different ecosystems, and we can see that the airborne data provides very powerful for like solar-induced fluorescence, different kinds of hyperspectral data, vegetation indices, spectral diversity index to monitor how the Earth is changing in response to the changing climate. But more efforts are needed to interpret the remotely sensed data. Some people may think, okay, we do remote sensing, so we sit in front of the desk, in front of my computer, and let the drone fly and get data for me. So I don't even need to go to the field. Well, that's not going to happen. So you need a lot of field work like this one here to interpret what is actually happening in your field, in your plot, to interpret the remote sensing data. Okay, so Socrates said this a few thousand years ago. He said that man must rise above the earth to the top of the atmosphere and beyond. For only thus will he fully understand the world in which he lives. Okay, now we have the chance. And I want to look at your plans in a different way. Come to fly with us. Thank you.
All right, great. Thanks, Rand. That was awesome. Yeah, if anyone has any questions here in the audience, or we can do some on Zoom as well, I'll bring the mic around and we can take questions.
Hello, Rand. Thanks for your talk. You mentioned early this idea that we need airborne data and you can't really do this with drones. Can you say a little bit more about why that is? Like, why can't you do the same thing with drones? Or what is the particular difference or value here that is missing from other platforms? Because we all like drones, right? They're cool. Can't they do this?
Yeah, so in this book, I think it's so in this book there are a whole section discuss the drones. Actually, so I like drones, and maybe fly drones I think is easier, much cheaper, and less stress than flying an airplane. But there are some things you need to consider. Like one thing is, so the typical drones can only carry a certain amount of instrument, and they can only fly for a certain amount of time. Let's say you have a drone, you buy a drone, and you can fly 30 minutes, 40 minutes. That's for one battery. That's very good. And how much you can put on that drone? Like maybe 20 kilos. But for this instrument, the IBIS here is a huge one. I don't think a typical drone can carry this one easily. And also for the airplane, if you want to get data, like the data here for different size, you can possibly cover all the size or most of the size if you plan well within maybe 1 or 2 days. But for drones, there's no way to do this one. And in this book, they discuss that the drones are good for certain things at the fine scale, at the local scale. But if you want a drone to fly to cover larger areas, to have a long time, like fly 4 hours in the air, and to cover a huge area and to carry, let's say, 100 kilos instrument. It's not possible for drones. That's a particular power of the airplane, I think. Yep. Yes. So
I know the Flex Tower's out there, but they have a couple of pods, version 3 out there. I was wondering if you did any work to relate that back to the sensors.
Yes, we actually have done this. We have done a lot of work with the CSD3 data. So we have the flux data. So the the blue line here in this figure, that's the GPP calculated from flux data at equals, and they give those data to us. And we have the ground safe data mounted on the scaffolding. And we have the airborne data for safe, NDVI, and some other indices. The first thing we did, we spent almost 2 years to try to generate these products first, because the safe calculation or the safe retrieval from the airborne data is actually not a trivial thing. It's very complicated. So we get that sorted out, I think. So the next step is we put everything together, combine the ground data and the airborne data to do more analysis. And actually, so John is working on a paper, so using all this data and putting them together. Oh no, not that one. So we haven't used that data. So we mainly focus on the airborne and also the ground safe and the flux data. That's what we have.
How easy is it to scale this up to like continents or regions and that kind of stuff?
Yeah, that's a good question. So actually, we did some of that work. Like, let's say in this figure, it's kind of show the idea of scaling or do the remote sensing at different scales. When you have a drone, you get fine scale data, like a very tiny but very high spatial resolution, like a centimeter or 10 centimeter. If you get the ground data, you have a higher resolution. And if you have an aircraft, you can do regional coverage. And you have satellite, it's covered the global, but with cost, spatial resolution. So I think it depends on what your research question is. It's really hard to say, is there a good or the best instrument you need to do? And a better way, I think, is like what do you want to accomplish with the data? And also the scaling question from like one single leaf to the global. There are a lot of things to think about, like in the case of safe, you need to think about from the leaf level safe, then you go to the canopy. So it's different. The canopy can reabsorb some of those safe signals. So technically, it's a sort of energy in that red region, right? So the plants can still absorb that. Then from the canopy, you need to think about the canopy structure. Then you go to a larger scale, then you need to blend in different landscapes like land cover types. You have different crop types. You have maybe one single pixel. You have different types of landscape. So that's a mixed pixel problem. And in the global scale, in the satellite, you have unique problems to deal with the satellite data. So yeah, I do think that's a great question, but I don't think there's an easy answer to this question. So the good way to answer is when we combine everything together, it probably gives us a better solution for the questions you want to do or want to work on. Thanks. Yep.
Have you thought about SAR, especially with mixed pixel issues and like semantic classifications, especially in canopy classification? I come from a mangrove background, and SAR is extremely popular, and the resolution, of course, is way better than anything like Sentinel Sentinel-1 is way better than obviously Sentinel-2.
Yeah, but that's still at larger scales, and also, for like the SAR image, they have some good parts they can provide, like the soil and then the soil water, and we use that data a little bit, but not a lot. So for this work, we focus on the optical remote sensing part without worrying too much about the active remote sensing. And the one thing we are thinking about is using LiDAR. So in the future, but for SAR, we just use some mature products provided by NASA or the ESA. So we haven't done any actual analysis using those data.
I'll just check Zoom if there's any questions. Nothing. Okay, well, we're just about out of time anyway, so maybe we'll just give Rand another round of applause and call it good. Thanks a lot, Rand.
Thanks.
Thank you.